There is grandeur in this view of life.
Welcome to Evolution Talk with Rick Coast, an introduction to the oldest story ever told.
This portion of your Mickey Mouse Club.
Hey, everybody, it's me, Mickey Mouse.
You're not really afraid of little me, are you?
Our relationship with mice is one of fascination and a little bit of fear. My wife is deathly afraid of mice, but she loved the mouse in Stuart Little and the rat in Ratatouille. And who didn't get a kick out of Tom and Jerry as a kid? But of course the cute ones are the animated ones, the cartoons. Wake up in the morning to the sound of little feet scurrying across your bedroom floor, and it's time to set the mousetrap. Rest assured that no mice were harmed during the making of this episode, at least not as a result of this episode. There are mice out there who might have a few things to say about their current status in the animal kingdom. We might love the animated mice on both the big screen and the little screen, but nobody adored mice like Abby Lathrop did. I'm talking about the real flesh and blood mouse. Abby was born in 1868, and her parents were school teachers from Granby, Massachusetts. It's a small farming town. It's still small, and it still has working farms. And it boasts a very small community still of a little more than 6,000 people. In Abby's day, it was even smaller. She became a school teacher herself, just like her parents, but poor health pushed her back to Granby, and she purchased her own farm. Not particularly keen on farming, Abby decided she would instead raise something. She liked mice, and this coincided perfectly with the trends of the time. Mice were all the rage. They were called fancy mice because of the ability of breeders to develop mice of different colors and sizes. And at the turn of the century, people were keeping fancy mice as pets. For Abby, business was booming, and at one point her farm boasted upwards of 11,000 mice. And to keep track of it all, Abby kept meticulous breeding notes. When scientific researchers came calling, Abby had more than enough mice, as well as records, to satisfy them. Things moved along nicely, until Abby found skin lesions on some of her mice. She was of course concerned, so she turned to some of the scientists that she had met along the way to look at the mice. One scientist, the pathologist Leo Loeb, told her that the skin lesions were malignant, and thus began a long collaboration between the two. Abby had more than enough mice for study, and Loeb had more than enough mice to study on, especially when it came to cancer research. And as a result of their collaboration, cancer research was pushed forward a few years, and the two of them co-authored articles that appeared in journals, and one such journal being the Journal of Cancer Research. And not bad for a retired school teacher living on a little farm in Granby, Massachusetts.
I profess to learn and teach anatomy, not from books, but from dissections, not from the tenets of philosophers, but from the fabric of nature.
That was English physician William Harvey. Harvey was one of the first to use mice for research, and this was back in the 16th century. Harvey was fascinated with blood circulation and reproduction, and he saw in his furry little friends the perfect opportunity for study. The English philosopher Robert Hooke also used mice for research. This practice continued, and in the 18th century, philosopher, theologian, and chemist Joseph Priestley also saw the benefits in using mice as research assistants. Priestley is even credited as being the discoverer of oxygen. And not that he had to go very far to find it, since he was pretty much surrounded by it.
I took a quantity of air, made thoroughly noxious by mice breathing and dying in it, and divided it into two parts, one of which I put into a phial immersed in water, and to the other, which was contained in a glass jar standing in water, I put a sprig of mint,
And this was in 1771.
and after 8 or 9 days, I found that a mouse lived perfectly well in the part of the air in which the sprig of mint had grown, but died the moment it was put into the other part of the same original quantity of air, and which I had kept in the very same exposure, but without any plant growing in it.
Those thoughts were recorded in Priestley's 1772 work, Observations in Different Kinds of Air. Gregor Mendel, the famous friar and father of modern genetics, also started his work on inheritance by using mice. He looked at coat colors, but he had to switch gears when his superiors asked him to concentrate on something a little more cleaner than mice. So he switched to peas. Almost decades after Mendel's death, the French biologist Lucien Quinault picked up Mendel's studies on mice and showed a skeptical world that Mendel's pea experiments on inheritance applied to animals as well. And this was in 1902, at the same time that mice were becoming a household pet. It could be looked at as the pinnacle moment for mice. Now, if you've ever thought to wonder why mice have been and continue to be science's favorite research tools, it's because of this. We are a lot alike. Yes, that little four-legged furry bundle of whiskers and pink feet shares 99% of its genes with you. And to top it off, those genes are also synthetic. Synthetic means that they, the genes, are often in the same order as they are when compared to our genetic code. These ancestral repeats are there because they were inserted into our genome before we split off. The next time you look at a mouse, just to know that you are carrying most of its 30,000 genes in your makeup. 99% of them. 75 to 80 million years ago, that 99% was 100%. That was when our most recent common ancestor walked the earth. That ancestor split off into different directions, and one lineage led to us, and the other led to mice. Now, we've talked about this before, but there are coding and non-coding regions in your DNA. And by coding, I mean protein coding. Proteins give your cells and your body its structure and its function. Now, without protein, you would be nothing at all, actually. 2% of your genes are protein-coding genes. The rest, the non-coding regions of DNA whose functions we aren't really sure about, those are called junk DNA. And I bring this up because our protein-coding regions, when compared to that of mice, have so many similarities in both order and function that it boggles the mind when you're made aware of it. There are even similarities in the non-coding or those junk DNA sequences, and these similarities are much smaller, about 50%. It's those DNA regions that match where the real magic begins. Mice multiply, and they multiply fast. Forget about 9 months. They can do so in 9 weeks and produce 6 pups each time. Now, if we assume that there are at least 3 females per litter, and each female produces 6 new pups every 9 weeks, well, math really wasn't my strongest suit, so I'll let you do the math if you want to. But to save us both the trouble, let's just say that at the end of the year, you will have a lot of mice running around. Just out of curiosity, I found a paper by Professor David Wharton from the University of New South Wales, and he figured this out. Take 2 healthy mice, leave them alone, and after a year, if it all works out, and we have a very productive brood, the total number could be over 5 million mice. Now, I'll link to this paper for those mathematicians out there. Now, I can't speak to its validity, but for everyone else, including myself, we'll just take Professor Wharton's word for it. Now, that's all assuming a year's worth of healthy mice. Not all mice are healthy, of course. Remember those lesions on Abbie Lathrop's mice? Mice can develop some of the same afflictions that we do, and this includes cancer, diabetes, and a host of other diseases. In part, this is why science loves to use them for research. That and their ability to refresh their population every 9 weeks. They also don't cost a lot to take care of. But the real reason that mice are so popular in the eyes of science is that we can manipulate the mouse genome to produce diseases they don't naturally get, like Alzheimer's. Setting ethical concerns aside for the moment, the thing is, because their genome is so much like ours, it makes using them for research incredibly appealing. Researchers are able to breed mice with genes similar to those prone to human diseases. I mentioned Alzheimer's, but they can do the same for cystic fibrosis and even Down syndrome. These are called transgenic mice. They being the researchers, they can also turn genes off to mimic what happens when mice come into contact with external pathogens or carcinogens. These are called knockout mice, meaning that the genes being targeted are knocked out of commission. By far, the favorite breed to test with is from the species Mus musculus. These are the same ones you might see run across your kitchen. Those bred for research are done specifically so, and they are kept under strict control conditions. Now, it's because of these strict methods that one of the most popular strains used, known as C57BL/6J, can trace its lineage back to Abbie Lathrop's mice. Now, we're talking 80 years here. They are so popular that they make up 83% of mice used for research in the US. And it was this very strain that had its entire genome sequenced in 2002. Now, so I don't have to keep repeating the entire alphanumeric combination, I'll refer to them as Abbie's mice. Just know I'm talking about C57BL/6. So, Abbie's mice are dark brown. They're very sensitive to noise, pain, and cold temperatures. And they also like to drink. And when I say drink, I do mean alcohol. They're very prone to addiction. Now, because they're so sensitive, Abbie's mice agitate easily and they are slightly more aggressive than other strains. A more docile strain, and also popular, is the BALB/C strain. These are those cute little albino mice that you might have seen in cages. These little guys are very useful for cardiovascular research, and they're very anxious. And then there are the mutant strains, scientifically created mutant strains. There are strains of transgenic mice. Remember, transgenic mice means that their genes have been tinkered with to be more susceptible to the same diseases we are. There are transgenic strains which produce mice that are larger than normal, those with better memories too, and also regenerative capabilities. And there's also one strain called waltzing mice, not because they like to dance, but because they have an inner ear condition which causes them to continuously walk around in circles.
The mouse model is becoming one of the core research tools in all of medicine, and including in cancer. And that has to do with the fact that we can actually engineer the mouse in remarkable ways.
That was Dr. Edison Liu. He's the current CEO of the Jackson Laboratory and President Emeritus of the Human Genome Organization. At the time, he was speaking at the WIN conference in 2012. The home base of the Jackson Lab is located in Bar Harbor, Maine. That's just a little over 3 hours away from where I'm sitting right now. It's one of the largest sources of laboratory mice in the world. It was founded in 1929 by Clarence Cook Little. Little developed the C57BL strain that's so popular today. So what about the treatment of those mice? Who's looking out for them? In both the UK and the US, there are regulatory bodies set up specifically to do so. In the UK, there's the Home Office under the Animals Act of 1986. Every year they publish a report on the use of lab mice, and the regulations themselves are pretty strict. One figure I came across is the number of regulated procedures. That number was over 3 million. I can't imagine what the size of that regulation book is. And here in the U.S., we have the Office of Laboratory Animal Welfare, which is tasked with overseeing the Public Health Service Act. Now, one of their duties is to make sure that any research project that hopes to receive government funding is compliant with the PHS Act. So where does all of this leave us? It leaves us with this. We have, as a species, overcome quite a lot. A large portion of the diseases that haunted us in the past are either under control or have been completely wiped out. But we have a long way to go. Whether it be Alzheimer's, heart disease, cancer, or Down syndrome, many of the clues and the possible cures are hidden in our DNA. A few flips of the genetic switches here and there, and it's possible that we may be able to see a future free from these diseases and afflictions. We can see that future with the same glasses we use to look into the genome of our furry little cousins. The thing is, once we find the keys, where do we stop? How many doors will we open? We can already create mice with human livers to research diseases like hepatitis, and since 1998, we can now clone mice. What if we were to develop a strain of mice with a human organ, or with a different human organ than a liver, like a heart, or a brain? Now, I'm stepping into science fiction here, but that's what we once thought about going to the moon, that it was all science fiction, or the creation of bionic limbs and even eyes. And just recently, in the past couple of weeks as a matter of fact, I read an article that talked about connecting the brains of 4 rats to each other and to a computer. The researchers were able to synchronize the rat's brain activity, and by doing so, they in essence created a small biological computer. The rats can theoretically work together to solve a problem with a brain network.
The billions, perhaps trillions of Borg drones operate as a hive mind. All thoughts are only those of the collective.
The Borg have a collective consciousness. There are no individuals.
We are the Borg. You will be assimilated. Resistance is futile.
I would like to thank Mike Willis, whose email inspired today's show. Evolution Talk is written and produced by myself, Rick Coast. The music on this episode was composed and performed by Poddington Bear. Support for Evolution Talk comes from you, simply by listening. Your support is greatly appreciated. And you can always reach me by using the site's contact page at evolutiontalk.com or on Twitter, where you can find me at Rick Coast. And most of all, thank you for listening. And until next time, please take care of yourself.
Mice? What do you mean, mice? I think we must be talking at cross purposes.
These creatures you call mice are not quite as they appear. They are merely the protrusions into our dimension of vast, hyper-intelligent, pan-dimensional beings. The whole business with the cheese and the squeaking is just a front. A front? Oh, yes. You see, the mice set up the whole Earth business as an epic experiment in behavioral psychology, in which your planet Earth and its people formed the matrix of an organic computer.
Evolution Talk is a Rick Coast production.